in approaches to drug delivery. My name is Hector.
My name is Richa. My name is Melissa.
And I'm Noah. And as for drug delivery, as of now, there are a few problems that we've seen. We separated them into two categories, in vivo and in vitro. So for in vivo drug delivery, the main issue we saw was toxicity in the whole system of the body because we're not targeting specific tissues and areas of the body that need the treatment.
For in vitro, the drug testing concerns include the inability of animal models to mimic human diseases, and drugs are not always safe when went from animal trials to clinical trials. So to start off, in vitro models, there's organs on a chip cell culture devices which recreate the microenvironment of the lung, liver, gut, kidney, and many other organs. Today we'll be talking about specifically pulmonary edema and hematopoietic disorder.
And another in vitro model is the cell culture system to monitor atherosclerosis.
So to start off, we'll be talking about pulmonary edema. Pulmonary edema is a condition caused by excess fluid in the lungs. And this fluid collects in many air sacs in the lungs, making it difficult for the person to breathe. So the device models 3 layers. The first top layer has alveolar capillary interface, which has air pass through it. In the middle and second layer is epithelial cells, and the bottom are the capillary cells through which blood is flowing through, and that's shown in the first diagram. In order to model the human pulmonary edema, IL-2 cancer drug was infused through the vascular channel, which causes shift of fluid into the air sacs and caused blood clot formation in air spaces and decrease in oxygen transport. This was measured through vascular leakage measure, as shown by the green dots flowing from capillary to lung. This ability of the chip to model this disease emphasized that it could be used to find a new therapeutic that can suppress IL-2-induced pulmonary edema. Next is hematopoiesis. Hematopoiesis is the production of all types of blood cells, which include formation, development, and differentiation of blood cells. In order to model the living bone marrow, PDMS devices containing cylindrical chamber filled with bone marrow producing materials were implanted on the back of a mouse and then surgically removed. Then the surgically removed implant was put into a similarly shaped central chamber in a microfluidic device. Then fluorescence activated cell sorting analysis illustrated that the engineered marrow had a hematopoietic niche similar to natural marrow. As a result, and not only this, because of the model produced living bone network identical to natural bone, the bone marrow on a chip will be used as a tool to study bone matrix remodeling in the future.
My research focuses on the in vitro cell culture system, also known as the stress angle device. We try to detect atherosclerosis at its earliest stages in the process, and just to give you a little bit of background, atherosclerosis is the buildup of fatty plaques in the walls of the arteries, and it is also known as the silent killer because it is not apparent, the symptoms are not apparent until later stages in the disease. Therefore, it is vital to understand the progress of this disease in order to prevent and predict adverse health issues by this disease. Now, previous researchers have investigated that endothelial cell cultures, endothelial cells are affected by forces like fluid shear stress and cyclic circumferential stretch, which is caused by the arteries stretching as the heart beats. In areas susceptible to the disease, endothelial cells respond to these forces by changing the orientation, proteins, and structure. Now, we developed and designed an in vitro device for mechanical stimulation of vascular cells and tissues in an effort to better understand the response to in vivo physiologic mechanical conditions. Now, for this research, this device, as you can see here, is capable of applying simultaneous fluid shear stress and cyclic circumferential stretch to endothelial cells, and we are able to alter the orientation of the shear stress and stretch on the culture cells. Now, our goal is to investigate cell response to this condition through imaging and applying biochemical techniques to study the mechanical risk factors and the progression of atherosclerosis. Now, these are two examples of the fluid shear stress at the bottom and the cyclic circumferential stretch at the top. That is what occurs in the arteries.
Now, Noah and I are going to discuss in vivo approaches to drug delivery, specifically using nanoparticles and BMENs. And the main focuses are to relieve stress on the body by targeting only the tissues that need treatment, making them customizable for patients, and most in vivo approaches are on the nano and micro scale. My research focused on using nanoparticles as drug delivery devices specifically for cancer therapies. And you can change the slide, thanks. What's really cool about them is they're actually being used in cancer theranostics, so not only treating cancer tumors, but also diagnosing them and finding them in the body. And they're reducing drug toxicity in non-cancerous tumors. So most drugs have a non-specific toxicity, and therefore they cause damage to whatever tissues they come across. And nanoparticles are doing this by being responsive to tumors. So whenever they find a tumor that generally has a lower pH or a higher temperature, they then release the drug only inside cancerous tissues. The main focuses are in improving the coatings by actually using natural cell membranes, which allow the nanoparticles to stay in circulation longer, thus increasing their blood half-life. So the longer that they stay in circulation, the more likely they are to accumulate in cancerous tissues. And another focus is increasing the drug load. The main drug that is being tested with nanoparticles is doxorubicin. And so the more of the medication that you can fit on a nanoparticle, the more effective it is in treatment. And another interesting subset of the research is magnetic nanoparticles, which are actually being used for hyperthermia treatments. So once they come across the tumor, they actually heat up, causing apoptosis and effectively killing the cancerous tissue.
And BMENs are another potential solution to our in vivo drug delivery problem. And BMEN stands for biomedical electromechanical systems. And it is a mouthful, but it means exactly what it says. They're just small mechanical electrical systems that you can insert into the body to do a specific task. Most of them are on the micro or nano scale, so these things are absolutely tiny. And because of that, they have to be manufactured through photolithography, which you can see right here, where a substrate is laid down, photoresistant material is laid on top, and a design is placed on a mask. That is then placed on top, and then it's all bombarded with UV light, and all that's left is the design in the photoresistant material. There are some setbacks with this kind of technology, such as material choices. What may be easy to produce and manufacture isn't exactly biocompatible or easy to acquire. And some of the fabrication techniques aren't all-encompassing and as versatile as they need to be at the moment. But despite these setbacks, there are some devices that are in trials right now that are successful. And some applications that you can see, the size essentially determines where and how it's input into the body. For devices less than 200 micrometers, they can be injected directly into the tissue. And for less than 100 micrometers, they can be inhaled directly into the body. And here you can see some microneedles that were developed. They can deliver drugs through the skin where there might be damage otherwise. Actuators are developed, which are input-sensitive release switches essentially. Reservoirs to hold drugs have been produced, and electromagnetic arrays to manipulate magnetic particles are also being produced.
To summarize the future application, in vitro models, right here, dressed in red, build new ways to test drugs, and in vivo models, dressed in blue, are targeting single organ system for toxic drugs.
Are there any questions?